Good day, everyone. I myself, Gary O. Martinez, together with my group mates, John Christian Agustin, Jaira Grace Destacamento, Alfie Jane Omar, Romelet Coquilla, and Jihan Canillas, students from the class of BS Ed Science 2A, we are going to unlock new informations and new ideas regarding lipid metabolism. But before we proceed to our discussion, let me present to you first the objectives of this lesson. By the end of lecture, the students should identify the importance of lipids in the food. Next, they will be able to describe digestion of different types of lipids. Next, they will be able to illustrate absorption of lipids from intestine. And lastly, they are able to summarize transport of dietary lipids from intestine. Lipids. We all know that lipids is any of various organic compounds that are insoluble in water. They include fats, waxes, oils, hormones, and also certain components of membranes and function as energy storage molecule and chemical messenger. So, a lipid is a very important component of living cells because they contribute to some of the body's most vital processes. And lipid are sufficient source of energy where every grams of fat supplies 9.1 kilocalories, in which we all know that calorie is the unit for energy. So every excess energy from food is stored as adipose tissue in the body, and fats in the body are critical for storing energy. So there are minimal amount of fat which is essential in our food. First, to produce essential fatty acids. So we all know that essential fatty acids cannot be produced in the body. So they need to be consumed. So the function placed by essential fatty acids is to have a proper development and functioning of the brain and nervous system. Also, the essential fatty acid is responsible for regulating blood pressure, blood viscosity, uh, vasoconstriction, immune and inflammatory responses. And there are also minimal amount of fat which is essential in helping fat soluble vitamins for absorption. It Fat soluble vitamins like vitamin A, vitamin D, vitamin E, and vitamin K will be absorbed better when fat is consumed with them. There are three processes involved in the digestion of lipids. First one is the digestion of triglycerides. Next is the digestion of phospholipids. And lastly is the digestion of cholesterol esters.
Digestion of triglycerides. So digestion of triglycerides or the digestion of lipids, it is a process that involves breaking down of molecules or lipid molecules into smaller molecules or smaller lipid molecules. So digestion happens so that these molecules that break down can be absorbed into blood. So as you can see in the picture, there is the illustration of the chemical formula of glycerol and triaglycerol. So we know that the triacylglycerol is composed or consists of glycerol and fatty acids. So of course, when this triacylglycerol undergo digestion, it will break down into glycerol and fatty acids. And digestion of triglycerides can be digested through the process of emulsification and enzymatic hydrolysis by lipase enzymes. So let's proceed. So let us first know or tackle emulsification. or the first process of digestion of triglycerides or lipids. So when we say emulsification, it is the breakdown of large fat globule into small ones. So first, as you can see in the illustration or in the picture, it is still a large fat globule or it's still big. Then due to the emulsification, it it is dispersed into smaller molecules. So emulsification happens because of the bile. So we know that bile, it is an acid that can only be released by the gallbladder. So emulsification first happen in the small intestine. So let us discuss it more further. So emulsification first occurs in mouth by chewing. So we know that our mouth contains saliva, and also with the help of chewing, it helps the food to it helps the food to break down into smaller particles or to be digested more easily later on. Then after chewing, the food will be swallowed, so it goes to into stomach by peristaltic contraction. So we know that also our stomach contains an acid, liquid. So this liquid helps the food to break down or soften much more, so that later on in our intestine, it will be digested easily. So after passing through in the stomach, it goes to into intestine by peristaltic movement. So we know that the intestine, there's a gallbladder that produces and releases bile. So this bile is an acid of, so by means of due to the acid, it helps the lipids molecules to break down or emulsify into smaller molecules. And after that, the molecules will be break down and easily absorb into our blood.
Lipase is an enzyme that breaks down triglycerides into free fatty acids and glycerol. Lipases are present in pancreatic secretion and are responsible for fat digestions. There are different types of lipase enzyme, namely lingual lipase, gastric lipase, pancreatic lipase, and intestinal lipase. The most active lipase is pancreatic lipase. Lingual lipase. Lingual lipase generates non-esterified fatty acid from dietary fats during oral processing by lipolysis. Lingual lipase in rodents has strong lipolytic activity and plays a critical role on oral detection of fat. The functional activity of lingual lipase during oral processing of high-fat foods in human remains poorly characterized. This enzyme released into the mouth along with the saliva catalyzes the first reaction in the digestion of dietary lipid, with diglycerides being the primary reaction product. However, due to the unique characteristic of lingual lipase, including a pH optimum of 4.5 to 5.4 and its ability to catalyze reaction without bile salt, the lipolytic activity continues through the stomach. The hydrolysis of the dietary fats is essential for fat absorption by the small intestine, as long-chain triglycerides cannot be absorbed, and as much as 30% of fat is hydrolyzed within 1 to 20 minutes of ingestion by lingual lipase alone. Gastric lipase. Gastric lipase is an acidic lipase secreted by the gastric chief cells in the expanded mucosa in the stomach. It has a pH optimum of 3 to 6. Gastric lipase together with lingual lipase comprise the two acidic lipase. These lipases, unlike alkaline lipases, do not require bile acid or co-lipase of optimal enzymatic activity. Acidic lipases make up 30% of lipid hydrolysis occurring during digestion in the human adult, with gastric lipase contributing the most of the two acidic lipases.
As you can see on the picture, we are discussing how pancreatic lipase breaks down the triglycerol. We know that triglycerol is composed of glycerol and free fatty acids. Pancreatic lipase, an enzyme which has the capability to split the fat or triglycerol, and this enzyme is released by the pancreas. This enzyme is very important for fat to complete digestion in the small intestine. The pancreatic enzyme breaks down the triglyceride into simpler compounds as monoglyceride and two free fatty acids. As the process continues of converting the triglyceride into simpler compounds for better absorption, the monoglyceride and two free fatty acids will become glycerol and two free fatty acids. Intestinal lipase act within intestinal mucosal cells, hydrolyze the absorbed primary monoglycerides, forming glycerol and FFA. Lipase, an enzyme the body uses to break down fats in food so they can be absorbed in the intestines. Lipase is produced in the pancreas, mouth, and stomach. Most people produce enough pancreatic lipase, but people with cystic fibrosis, Crohn's disease, and celiac disease may not have enough lipase to get the nutrition they need from food. So let's go now to the digestion of phospholipids. Phospholipids is converted to lysophosphides. Intestinal phospholipase may complete the hydrolysis of lysophosphides. Phospholipase activated by trypsin and requires bile salts for activity. The enzyme lingual lipase, along with a small amount of phospholipid as an emulsifier, initiates the process of digestion. These actions cause the fats to become more accessible to the digestive enzymes. As a result, the fats become tiny droplets and separate from the watery components.
Digestion of cholesterol esters. When we talk about cholesterol esters, it is mainly made up by cholesterol esterase, which can be produced into FA and free cholesterol. Next slide is about absorption of lipids. The absorption of lipids have an end product, which are the monoglycerides, FA, glycerol, cholesterol, and lysophospholipid. When we talk about glycerol and short-chain FA, it is always water-soluble, which can be carried through portal circulation. And when we talk about long-chain FA, monoglycerides, cholesterol, and lysophospholipids, these following need bile salts in order to be absorbed. Next slide is about bile salt. What is bile salts? Bile salts surround this component by their unpolar end while their polar endings directing outward, which are water-soluble mesocels 0.1 to 0.5 in diameter. In the diagram, there are some circles directing outward in the circle, while the center point is the lipid, and the circle that is gathered around the lipid, each one is called bile acid. And bile acid has two sides that are which are the hydrophobic side and the hydrophilic side. The hydrophobic side is the unpolar end of the bile salt or the bile acid, while the hydrophilic side is the polar end, which are directing outward of the bile acid or the bile salts.
So let us continue. So micelles are soluble in water. They enter microvilli or mucosal cells by endocytosis. So endocytosis is the process by which cells take in substances from outside of the cell by engulfing them in a vesicle. This occurs when a portion of the cell membrane folds in on itself, encircling extracellular fluid and various molecules or microorganisms. This is where fat digestion may be completed through reaction of intestinal lipase. In the mucosal cells, triglycerides and other lipids are resynthesized once again as follow. As you can see here, fatty acids was being activated to a acyl-CoA. So a carboxylic acid and a coenzyme with the help of synthesis of acyl-CoA, it turns to carboxylic acid and another coenzyme. Acyl-CoA is a group of coenzymes that metabolize fatty acids and are susceptible to beta oxidation, forming ultimately acetyl-CoA. The acetyl-CoA enters the citric acid cycle, eventually forming several equivalents of ATP. So as you can see here, in this way, fats are converted to ATP, the universal biochemical energy carrier. So fatty acids are activated by reaction with CoA to form fatty acyl-CoA. The reaction normally occurs in the endoplasmic reticulum or the outer mitochondrial membrane. This is an ATP reaction yielding AMP and pyrophosphate. So that was it. And second is reesterification of absorbed beta monoglyceride with 2-acyl-CoA will result to triglycerides. Reesterification of glycerol 3P with 3-acyl-CoA will result to triglycerides. And glycerol 3P is derived from glycerol by glycokinase present in liver, kidney, and to small degree in small intestine but deficient in adipose tissue. DHAP derived from glucose by glycolysis. The synthesis of phospholipids and cholesterol esters by combination of cholesterol and glyphospholipids absorbed with acyl-CoA. The triglycerides, phospholipids, and cholesterol bind with a protein called apolipoprotein B48, forming chylomicrons to lacteals and pass with lymphatic drainage to the thoracic duct systemic circulation. So let's move on to the fate of absorbed lipids. So immediately after absorption of lipids, there is turbidity of plasma due to circulating chylomicrons. And these appear in plasma 2 hours after meals. So what is chylomicrons? These are large triglyceride-rich lipoproteins produced in enterocytes from dietary lipids, namely fatty acids and cholesterols. Chylomicrons are composed of a main central lipid core that consists primarily of the triglycerides. However, like other lipoproteins, they carry esterified cholesterols and phospholipids. So, this turbidity is uncleared by lipoprotein lipase enzyme, which is known as our clearing factor. Then after that, lipoprotein lipase to chylomicrons, then glycerol FFA, which means free fatty acids, and then lastly, glycerol to liver, and free fatty acids to adipose CT liver.